I am now, as Susan said, an assistant professor in biomedical engineering, and I actually run a joint lab with my postdoc advisor, Chris Schaefer. And actually, I'll tell you a little bit more about this, but this work is actually done in our joint lab and is actually the product of a long collaboration. So today I'm going to share with you a story of science. And it's about optical tools and what they can do in biology and how some of the technologies that we've developed in optics and physics actually translate to a lot of different other topics. In this case, when we're talking about blood flow in the brain and Alzheimer's disease. Okay, so let me get started by telling you what I think we need to be able to understand the biology of living systems. So living systems, like ourselves, are very complicated, but a lot of the fundamental interactions and fundamental dynamics are at the cellular scale, and the cells are on the micron scale, so something that's in microscopy, we require microscopy. But in the living body, one of the most important things to think about is that there are many different types of cells that are in constant communication, and their interactions and their dynamics actually determines what happens in the living animal, in normal function, as well as in disease. So what we need to make progress on a lot of the tough biomedical questions that are out there are tools that can get at this scale of a single cell, but also deal with the fact that it's not just one cell. It's a whole sort of community of lots of different cells that are interacting and competing in some cases, cooperating in other cases. So I'm interested in neuroscience primarily. And one of the things that we realized is that the brain is actually a quite homogeneous big lump of tissue. To be able to understand this, you really need to be able to go inside of the brain. And turns out that optics is a great way to do this. So this is actually a slice of rat brain, and you can see the blood vessels stained here. And this is about 2 millimeters deep. But one of the things that is a big challenge is that we really need to understand what's going on, not just at the very surface, but down in the depths of the tissue. So the way we dealt with that and the way we get to being able to study the cells in a living system is through nonlinear optics. So the field of nonlinear optics actually has to do with extremely high intensity, very powerful lasers. And the whole principle behind this is that when you have very high intensity lasers, you can pack them into such a state that you can get lots of photons to start interacting with each other. And then you can get things like fluorescence that I'll show you, and manipulation that's not possible, and you can go below the surface. But the basic principle is this. You want to get as many photons as you can so that you actually get a density at which they'll start to collide with each other and whatever other material that you have. So the key technology here is what's called an ultrashort pulse laser, or sometimes a femtosecond laser. And what it does is it takes all the light and basically packages it into very short bursts of time. So here we're talking 100 femtoseconds, which is 10 to the minus 13 seconds, which is extremely short, sort of the ratio between a day and the age of the universe. So these lasers are special because they do pack all of these photons in time, but we also pack these photons down spatially. So we take a lens, not so visible here, but we take a lens and also just focus down the beam. And what happens when you focus down the beam is that you can get a roughly micrometer size spot where the photons are at such density that they can actually collide with each other, interact with each other, and change sort of the nature of how they interact with materials. So the way we do this, or the way we use this in biological questions is we use it for a novel kind of microscopy. So in a lot of biological systems, we can tag different cells, we can tag different proteins and things with a fluorescent dye. And here's an example of a vial of fluorescent dye. And with this high pulse laser, what you can do is arrange it so all of the photons from this laser beam on this side get focused down to one spot. And because we need a lot of these photons to interact all at once, you can really limit where any of these interactions happen to just one spot. In this case, you're just seeing a little bit of fluorescence right there from the dye, and it's right at the focus of this laser. And this is in contrast to an ordinary laser a CW laser, which is basically constantly streaming out photons all at once, is here we can see that the laser is focusing down, but you actually get light from the entire path of the laser. The way this translates into a way to image in a biological system is we take this laser, focus it down into a sample, and then scan it around a sample. And then because we know that any of the interactions are happening right at that focal place and nowhere else, we can basically scan it around and map out where things like fluorescence are. So we can use this as a way to look and map out on a very small scale, on a subcellular scale, where any of these fluorescent tags are. So I'm going to talk to you about how these tools are leading to some insights on Alzheimer's disease. So let me digress a little bit and tell you a little bit about Alzheimer's disease. You may already be familiar with this disease, unfortunately, because it is very common. In the coming years, it's predicted that it's going to be about 1 in 8 older Americans will have Alzheimer's disease. This is a picture of one of the first patients that was identified with this disease by a doctor about 100 years ago named Alzheimer. And what the toll of this disease is that it basically destroys a person's ability to have a personality and to have their memories. But it tends to preserve their health. So there's going to be a huge number of older Americans whose personalities and their cognitive abilities are degenerating while they're still otherwise physically healthy. And in addition to the vast emotional toll that this is going to take on us, it's also going to be a huge financial burden for the health system. So one of the things that's very tricky about Alzheimer's disease is it's related to a process that happens all the time, even in a healthy person. So in Alzheimer's disease, what happens is that there's a small snippet of proteins called A-beta that accumulates in the brain. But A-beta is something that is made throughout a person's life. If you're awake and functioning normally, you're actually making this protein in quite huge quantities inside the brain. So it's a little bit hard to treat as a disease because the fundamental protein that's involved is part of normal function. In some people, what happens as they get older is that the protein starts to aggregate and it starts to accumulate in the brain. And right now, we don't really know why this happens to some patients and not to others, and why this all of a sudden, or actually not all of a sudden, over time becomes a disease that takes away cognitive abilities in some people but not others. So one of the key questions is, what are the triggers of this disease? So one of the hints comes from the clinical data that's out there. So it turns out that there are the related risk factors in between vascular disease and the accumulation of this protein and the dementia and Alzheimer's disease. So if you have high blood pressure, if you have diabetes, or if you are obese, your risk for Alzheimer's goes up. And these risk factors are things that we consider to be cardiovascular risk factors. So they're things that affect the blood vessels. So here was a hint that there's a relationship in between blood vessels and this brain disease. There's other interesting factoids that point to a relationship in between blood vessels and Alzheimer's disease. And one of the interesting phenomena that's been known for a very long time is that patients that have Alzheimer's disease, they actually have less blood flow to the brain. It's about 20 to 30% less blood flow to the brain than someone of the same age that doesn't have that disease. So to study this, we wanted to look in the brain of animal models, and we used these nonlinear optical tools, including multiphoton microscopy, to let us take a look and see what is going on. So mice don't naturally get Alzheimer's disease. So one of the technologies that we do use is transgenic mice. And they have genes that have been associated with families that get early onset Alzheimer's disease. And although our multiphoton microscopy enables us to look deep, it doesn't quite let us go through a whole mouse. So what we actually do is we implant cranial windows, basically a piece of glass right in the mouse's head, and then we can use this as a window to look and do microscopy. The mice actually do very, very well with these windows. We can implant them and we can have them around for up to half a year, and given that a mouse only lives a couple years, this enables us to observe a mouse and see what happens to the disease over a good part of their lifespan. So this is what multiphoton microscopy looks like in a mouse. So this movie takes us through the top layers of the mouse brain, and what we're seeing here is blood vessels. So the blood vessels are highlighted in white, and all these very fine hair-like, spaghetti-like features, those are the finest capillaries in the brain. So these are what carries the red blood cells and the blood through the brain. At this level, at the capillary level, you're basically getting one red blood cell at a time. So what this movie is doing is it's stepping from sort of the top of the cortex down into the mouse brain. And as long as, in addition to all the vessels, what you can see is occasionally a red sort of rosette. And those are the deposits of this A-beta protein that has been implicated in Alzheimer's disease. So if you look very closely at some of these blood vessels, and here's a movie where we kind of zoomed in, what you can see is that there's black and white stripes that go through any of these, most of these blood vessels. And what that is that you're actually seeing a red blood cell moving through the vasculature. There's dye in there, so the red blood cell doesn't take up the dye, so it basically occludes the dye, and you can see sort of the shadow of the red blood cell going by. And when we started this work, one of our students actually noticed that in a healthy brain, most of these vessels are flowing and flickering like this, so you can actually see the motion. But she noticed that in a few blood vessels in the brains of these mice that have this Alzheimer's disease, she noticed something like this. And here what you see is there's a shadow of a cell that seems to be stuck, and it's not moving. And turns out that there are lots of these stalled vessels in the brains of these mice. So after her observation, we actually launched a study, and what we found was that about 2% of the blood vessels in the brains of these Alzheimer's mice at any given time are plugged by a white blood cell that is basically completely plugging this capillary. In a normal mouse, and these are siblings of the mice that don't have Alzheimer's disease, there's basically no capillaries that are stuck like this. So the next step was, this is an interesting phenomenon, but 2% isn't that many, right? Like 2 in 100 vessels. So one of the questions was, how does this actually affect blood flow in a way that might matter for a human patient? So we actually turned to another tool that is also based on these high-power lasers. Here, this is femtosecond laser ablation with an extremely high-power laser that can actually ionize material. So this version of the femtosecond ultrashort pulse laser has been used a lot in electrical engineering, physics labs, and also a basis in the telecom industry for some of the things like trying to carry data on optical cables and that sort of thing. But here we used some of the physics that we had learned about how these femtosecond lasers interact with material to manipulate biology to try to mimic the disease state. So our goal was to understand why or what happens when one of these little tiny microscopic capillaries actually clots. And that's not something that you can actually do with any sort of surgical technique with your hands. So what we did is we took the laser that was used for in basically a physics lab, and we focused into a blood vessel, and basically it causes a tiny bit of ionization, and it nicks the blood vessel and triggers the same clotting cascade that you would have if you cut your hand. And this is what it looks like in a movie. So this is a blood vessel, a capillary, and what we're doing is we're focusing a femtosecond high-powered laser right about in the middle. And what you can see is that we're gradually making this vessel leak a little bit more, and then with a little bit more leakage, a clot actually forms, and that's this clot right here. And then when we zoom out, what you can see is that even though we've clotted this vessel and got a little bit of dye leakage, all the vessels around it are still completely intact, and that gives us the opportunity to understand and go study and see what's happening in these vessels. So we use this to answer the question of what happens when there is a clot in one of these tiny vessels. And so we mapped out sort of the speeds of blood flow in the vessels before and after there's a clot. And what we found is that when you clot one of these tiny vessels, it's not just that vessel, but it's the downstream vessels that are affected, and they're actually slowed quite considerably. And so it's not just those 2 downstream vessels, it's also the next 4 downstream vessels and the next 8 downstream vessels that are affected. And then if you put this back together, you can get a picture like this. So these are tracings of blood vessels in one of the mice that we've imaged. And here what we've done is we've marked all the vessels that we found are stalled. And so there's quite a few, but it's still pretty sparse. It's just about 2%. But if you start to consider the fact that the downstream vessels and the next downstream vessels and even those after are affected by this change in blood flow, you actually start to get the picture that there is actually a substantial flow change, and it can actually influence a very large region. And what we found in sort of our simulations and ongoing work in theory and also now experimentally is that this change in blood flow can be a change of blood flow about 20–30% to your entire brain. And if you experience that acutely, what would happen is that you'd probably feel a little bit woozy, and it would be definitely something that you would notice. So this work suggests that Alzheimer's patients, in addition to the effects of the A beta protein, are also probably experiencing a deficit in blood flow to their brains that may also be symptomatic. So our experimental results actually match very well to the clinical picture. That what we see in the mice in terms of how much blood flow reduction is actually matches quite well to what clinicians see in human patients. So one of the reasons this is exciting, though, is that there's a lot of groups that have discovered that changes in blood flow, especially if you decrease blood flow, actually leads to an increase in this A beta aggregation. And there's lots of different mechanisms for why that actually might happen. But what we've shown is that once you have this A beta aggregation, you also get a sort of a cyclical relationship where you get more of these blood vessels that are plugged and a decrease in blood flow. And what this suggests is that there's sort of a cycle that could get triggered by any event that might start some vascular inflammation, like all of these vascular risk factors like diabetes and hypertension. And this might be one of the things that leads to this accumulation starting in some people, and then that eventually leads to the disease of Alzheimer's disease. So this inquiry is sort of a new direction for a lot of Alzheimer's disease research, and it was sort of made possible by the fact that we had some new tools to be able to look at the level of the living cells inside an entire organism. So I just wanted to finish up with some of the most important things, and this is the people that actually did the work. So this is the list of people that have been involved in the work and are in what we like to call the Schaefer and Nishimura labs. It is a dual lab from my longtime partner, Chris Schaefer, and myself. Because this is about women in optics, I did want to talk a little bit about how I actually got here. For many women in academia and also women that are going into some of these business careers, women in science often end up in partnerships with their male colleagues or collaborators or that sort of thing. And more what you're getting is you're getting teams of people that are both in science. And if sort of as a science industry, as academia and an optics business, if we don't take advantage of that, we're going to lose a lot of opportunities. So for me, what that meant is I have worked and colluded with my partner Chris Schaefer for almost 20 years now, ever since I was an undergrad. And what that has led to now is a research lab that covers a huge span of topics and can invest in a wide diversity of tools, many of them that are optical tools. And it's been a great opportunity for me because we do run a joint effort, we can be the basis for lots of collaborations and look across many fields and bring together a lot of people that are all interested in neuroscience or biomedical problems. This work is actually also collaborative with a few people at the medical schools, and then people in, for example, in the applied engineering physics departments. But it is all supported by the fact that we're moving sort of towards team science and interdisciplinary science in many different ways. So with that, I just want to put up my acknowledgement slides for funding, and thank you very much for your time.